thank you everyone for coming. Jared Balbona is a statistician geneticist.
Sure. Geneticist.
From Matt Keller's group in the University of Colorado at Boulder. We've been working on a series of projects using the MOBA study to try and integrate some of the intergenerational genetics data that we've got. Jared's just about finished his PhD, was it?
Last month.
Last month. And as part of his PhD has not had the opportunity to, travel around the place as we did pre-pandemic. So it's fabulous to welcome you here and to show you Bristol and Manchester over the next couple of days. And I'm really looking forward to hearing about your project. So
Thank you.
Take it away.
I'm realizing I did not. Okay, so that's working there. And I just need to share my screen for Zoom, and then we'll be good to go. So hopefully that works. Hopefully everyone on Zoom is not getting my notes. That's embarrassing. Okay. Hey everybody, it's really good to be here. As Neil mentioned, I'm coming from the University of Colorado in Boulder. And I was just telling him that it's actually my first time ever leaving the US. So I'm very excited to be anywhere right now. So that's great. And I'll be talking about a project that I've been working on with Matt Keller and Young Yim for the past couple of years on estimating parental effects using structural equation models. And this is work that we're all really excited about. Hopefully we'll have some papers coming out soon on it. So without further ado.
Jared, can I just ask you to share your screen again? It's not coming through on the recording.
Sure. Is this any better?
That is perfect. Thanks.
Okay, great. All right. So to start, I think we've all had the experience where we meet a friend's or significant other's parents, and we think to ourselves, okay, yeah, that explains a lot. That makes a lot of sense. And that's a really understandable and really universal reaction, because from hobbies and careers to levels of extroversion and education, even things like mental and physical health and bodily mannerisms, there are a ton of ways that parents and children resemble one another, which is obviously nothing new, but still it begs the question of why. What is the underlying cause of all that parent-child similarity? And that question there is the overarching theme of my work, and it is the topic of this presentation. So in answering that question, we can start by looking at education level as an example, given that there is a lot of evidence showing that highly educated parents tend to also have highly educated offspring. So in thinking about why that may be, we can start with what's probably the most obvious explanation of parent-child similarity, which is shared genetic effects. So your mom and your dad transmit half of their additive genetic effects to you. And as a result, the same genetic factors giving your parents a predisposition toward a given trait are also going to be giving you a predisposition toward a given trait. So in other words, parents and children are phenotypically similar because they are genetically similar, which is simple enough, but maybe not the full picture. Instead, you can imagine an alternative scenario in which being highly educated itself is impacting how parents raise their child. So for example, a highly educated parent may be more likely to encourage a positive attitude towards schoolwork or to read to their child at night. Maybe they just have more books lying around the house. So all of these different behaviors that collectively could have an influence on the offspring's education level down the line. So in contrast to genetic transmission, this effect is known as vertical or sometimes cultural transmission. So with vertical transmission, the parental trait is influencing the offspring's familial environment. That's the environment where the child is raised, where they grow up. It's created and shaped by the parents. And that familial environment could in turn influence the offspring's trait later on. So vertical transmission is often an overlooked source of trait variation among humans. It's at the heart of the nature nurture debate, obviously, and it's crucial to understanding why we are the way that we are. But that said, it has historically been really difficult for researchers to distinguish this vertical transmission path here from this direct genetic path here, as both will lead to traits running together in families from one generation to the next. And it is especially difficult to distinguish the two when they co-occur, which will happen anytime that parental trait up there is itself heritable. So like a lot of the time, because pretty much everything is heritable. So note the circularity in this diagram. So the same genes that are directly influencing the offspring directly are also influencing the parental phenotype, which in turn influences the offspring via vertical transmission. So that means that children who inherit genes that are conducive to a given trait, in this case higher education, are more likely to also inherit an environment that predisposes to that trait as well. So when there's vertical transmission, the individual's genes and environment will not be independent of one another, but rather the two will co-vary, with this covariance being referred to as passive gene environment covariance, or more recently, genetic nurture. So like vertical transmission, the magnitude of genetic nurture carries a lot of important information about the importance of parenting and shaping who people become, and the mechanisms that underlie that process. So as such, genetic nurture is itself a really worthwhile thing for geneticists to be interested in looking at. But that said, even for researchers who are not specifically interested in parenting, genetic nurture and vertical transmission still have a number of really important consequences. So for instance, the occurrence of vertical transmission means that studies looking at the effect of a gene or a SNP on an outcome are not only capturing the actual direct effect of that gene or SNP on the outcome, but also the effect that it had on the person's parent, which in turn influenced the offspring via vertical transmission. So as a result, genetic nurture will lead to biased estimates of genetic effects across a variety of designs, including both GWAS and twin models. So sometimes this bias is upward, sometimes it's downward, depending on the specific model being used. But either way, therefore, I think it's generally safe to say that vertical transmission and genetic nurture are important constructs for us to be accurately and reliably estimated. So to that end, a number of recent papers, and most notably this 2018 paper from Kong et al., published in Science, have implemented a pretty novel and interesting approach. So specifically, instead of just looking at the genetic factors that are shared between parents and children, so that is the alleles that parents are transmitting to their kids, these researchers have also incorporated the portion of the parental genomes that is not shared between parents and offspring. So those alleles that the parents do not transmit to their child. So like the transmitted alleles, the non-transmitted alleles will still impact the parental phenotype, and thus they can still influence the offspring's phenotype via vertical transmission. However, unlike the transmitted alleles, that is the only impact that the non-transmitted alleles can have on the child. So without the familial environment and without this vertical transmission path there in the middle, there is no connection between the non-transmitted alleles and the child. They're independent of one another. So that means by essentially subtracting these paths in green from these paths in red, we can get an unbiased estimate of this direct genetic effect down here. And we can use that unbiased estimate to in turn estimate the effect of vertical transmission. So in contrast to previous designs, this approach was really significant in the amount of information it's able to provide using a relatively simple model. So unlike past designs, which required having adopted children and their adoptive parents and their biological parents, or having twins and their full extended families, this design just requires having data on mothers, fathers, and children. So a lot simpler. But nonetheless, like any approach, there still has its limitations. So these researchers first they assume no sortative mating beyond the parental generation. So their model cannot work for traits where the parents are sorting on the trait, or they have been sorting on it for an extended period of time. Despite having all the tools to do so, these authors didn't actually estimate vertical transmission itself. They were assuming that the polygenic score was accounting for full trait heritability, which is currently untrue for any existing polygenic scores. And finally, they derived a lot of their math from scratch in order to specifically look at the transmission of education level in Icelandic families. So that's really impressive to derive all that math from scratch, but it means that it mostly applies to studying education level among Icelandic families. So it can't easily be extended to other types of data or other phenotypes. So to address those limitations, Matt Young and I took their logic and instantiated it into a series of causal models that we are referring to as SEM-PGS, because they are structural equation models that incorporate polygenic scores. And SEM-PGS has a number of advantages over previous designs. So first, using only polygenic scores, it can provide unbiased estimates of heritability, vertical transmission, and genetic nurture. And it can do so even when the polygenic score being used is itself not very predictive. So our model's point estimates are not impacted by the predictive ability of the PGS. Secondly, it can properly account for various types of assortative mating at both equilibrium and disequilibrium, which I'll go over in more detail later. Third, in principle, it is not biased by missing data if those data are missing at random. So it doesn't require complete trios. If your data set just contains mother-child or father-child pairs, that works too. You'll just need a larger sample size to achieve equivalent power. And even if you just have a lot of spousal pairs, you can still get a lot of important information for our models from that. 4, it's relatively easy to use, relying on simple math, which I hope to demonstrate in this presentation. And 5, by virtue of being a structural equation model, it can allow for a vast number of extensions to best fit the trait and data being worked with. Okay, so before getting into the full structural equation model, and do people here have general familiarity with structural equation models, or are these? Okay, seems like a little bit, okay, great. So we can start here with the paternal genes and phenotype on the left and the maternal genes and phenotype on the right. So note the P and the M subscripts. So in the middle, we have these Ys for each person's phenotype. And for these squares, the observed variables, we have T's to represent polygenic scores made from the transmitted portions of the parental genotypes, and MTs to represent polygenic scores made from the non-transmitted portions of the parental genotypes. And because these transmitted polygenic scores are definitionally shared between parents and offspring, they will have the same impact on the offspring's trait that they're having on the parental trait. So there's the offspring's phenotype at the bottom, note the O subscript. Another way to put it, these NT and T polygenic scores add up together to be the parent's full PGS, whereas the TP and TM variables add up to the offspring's full PGS. Does that make general sense? Cool. And as some of you, if you do structural equation modeling, might have noticed, the parental phenotypes are represented by circles, meaning that they are not observed. You don't need to have observed parental phenotypes in our model, which is a really big advantage of using it. It's not important that the same questions are being asked to parents and offspring. All you need in the for the parents is whole genome data. You just need to have a measured phenotype for the offspring. Okay, so if we take this diagram and add on variances for each of the PGSs, path coefficients for each of the paths, and residuals for each of the phenotypes, we now have a complete structural equation model of each person's direct genetic effects, which of course is not in itself particularly informative. This model here is essentially just saying that human complex traits are functions of 2 things. 1, the person's genes, and 2, literally everything else, which is technically correct, but it's not telling us anything about genetic nurture, vertical transmission, or assortative mating. So to that end, we can add in these 3 latent F variables to represent each person's familial environment. So that's the environment in which the person is raised, the environment that is created and shaped by parents, And because it is created and shaped by parents, we'll have these two arrows here from the parental phenotypes to the offspring's environment. Okay, so finally, recall that people's genes and familial environments are often non-independent of one another in the case of genetic nurture. So we can capture that non-independence in this model by adding in covariances between the individual's genes and their rearing environments. So these covariances here on the sides are that genetic nurture covariance that I showed earlier. And they account for the portions of the polygenic scores that are tagging environmental effects and vice versa. Okay, so this model here is what we're referring to as SEM-PGS model 0. And by comparing variances and covariances among these 5 observed variables, we can get unbiased estimates of both genetic nurture and vertical transmission. And while this model does not specifically estimate or account for assortative mating, doing so is easy. But before I talk about that, any questions so far? Does this model make general sense?
Sorry, so you're assuming that the phenotype is observed in the offspring,
Yes.
but not the gene, or not necessarily the genotype. Is that right? Or are you going to use the offspring genotype?
You will need to use the offspring genotype to determine which alleles were transmitted.
Okay.
So our group is actually working on ways to figure that kind of thing out with a missing parental genotype, or well, yeah, for the time being, yes, you'll need to have a whole genome data for all 3 people.
Yeah, but not the phenotype.
Exactly. You can have that if you want, but you don't need it.
Is it absolutely essential to split off the transmitted and non-transmitted here to be able to identify them as a data point? Or could you merge the parental PGSs?
Do you mean combine these into one?
Yeah.
Well, then it would be necessary to split them off, because having an exact variable of just the alleles that are non-shared is what is providing that information to estimate vertical transmission. So that's kind of the crux of the whole thing, because on the assumption that this model is making, and assumption by Kong et al., is that the only connection that those alleles are having to the offspring is through vertical transmission. So that comparison between the two is what's giving you the information.
So yes, I guess there's 2 questions there. Firstly, if you're having to do that, don't you need to have both parents in order to identify the transmitted and non-transmitted?
You need to have their genetic data, but not their phenotypic data.
Okay, that makes sense.
Yeah. All good. Okay. And would it be helpful for me to give a quick primer on assortative mating before I go further? Is that something that people know very well here and study a lot, or? Okay.
Maybe a quick primer.
Yeah. Okay. Quick primer. Okay. So across many traits, people exhibit a tendency to mate with others who are similar to themselves, and that tendency is referred to as assortative mating. And there is widespread evidence of assortment across traits, which includes not only the literal thousands of correlations that are described in the literature, but also I'm sure everyone in this room can think of the couples that you know and the weird or important similarities that they have to one another. But despite all that evidence for assortment, for the sake of simplicity, geneticists often have to assume that all individuals in a given population are equally likely to mate with one another, regardless of their trait values. So in other words, we have to assume that coupling up just kind of happens with no systematic forces driving or shaping people's decisions, that it's all kind of just random. And that assumption is not only often going to be false, obviously, but it's unfortunate because of a couple of reasons. Namely, that assortment is just like a super cool thing in and of itself, which I know I'm biased about, but I think that it tells us a lot of information about how people are responding to shifting cultural landscapes and how their priorities are changing over time. So for example, as the door to higher education has become increasingly open to women in the past 100 years, maybe finding a highly educated partner is of greater importance to heterosexual men now than it was a century ago. Or we can look at how assortative mating patterns change during times of prosperity versus times of scarcity. So like how do priorities change during World 1 and 2 versus afterward. So I think assortment is really cool in its own right. But even if you don't care about that kind of sociological research at all, it's still important because it will impact the genetic and phenotypic architecture of traits, violating the assumptions of many models and biasing results. So specifically, assortment can lead to increases in population phenotypic and genotypic variance, homozygosity, genetic correlations between relatives, which violates the assumptions of a lot of traditional designs used in genetics, traditional family-based designs. Genetic correlations between traits. There's a recent paper by Richard Border that I think is under review at Science, detailing how a lot of the genetic correlations between traits that we've assumed to be evidence of shared genetic etiology are actually possibly just due largely to cross-trait assortative mating. So rather than there actually being shared causes for things like bipolar and schizophrenia, it could just be that people with those two traits tend to preferentially mate with one another. And 5, gameted phase disequilibrium, which is what I will be talking about now as it relates to our models. Okay, so to return back to the model I showed earlier, model 0. So here we're assuming no assortative mating, which means that each of these 4 parental PGSs will be independent of one another. So if we assume here on the right, with 2 loci from your dad and 2 from your mom, there will be no systematic connections between those 2. However, if there is a single generation of assortment, then these parental haplotypes will start to covary with one another. So for example, it could be that your father's height increasing alleles are correlating with your mother's height increasing alleles and vice versa. So our model can account for those with the addition of what's known as a copath between these parental phenotypes. So copaths are constructs used in structural equation modeling specifically for the purpose of studying assortment. So they're a very niche thing, but they work out very well for this one specific purpose. Okay, so to recap, after a single generation of assortment, there will be correlations between parental haplotypes. If there is more than one generation of assortment, then we will see those between haplotypic correlations, but we will also see within haplotype correlations. So it could instead be that your father's mother's height increasing alleles are now correlated with your father's height increasing alleles. It could be due to correlations in the great-grandparental or great-great-grandparental generation, depending on how long that trait's been assorted upon. And our model is able to account for those within haplotypic covariances, as well as the increased haplotypic variances with these green double-headed arrows shown here on the sides. So within each parent, we are now allowing for covariances between their 2 haplotypes, and we now are adding a G term to the variances for each of the PGSs. So this is what we're referring to as model 1. And while pretty simple, the addition of those covariances in that copath allow for us to account for a lot more relationships between our variables. So for example, now the dad's familial environment can correlate with the mom's non-transmitted alleles, can correlate with the mom's transmitted alleles, and so on and so forth. Everything can now be related to everything else. And the added complexity that you get from having to account for all of those different correlations between things is a major reason why past approaches have struggled to fully account for assortative mating. It can become mathematically intractable like really, really quickly. But fortunately, our model 1 that accounts for assortment is actually not that much more mathematically complex than our model 0. And I can demonstrate that by taking these 2 models that I've gone over. So one that accounts for assortative mating and one that does not account for assortative mating. And to show the difference between the 2 in practice, we can go over how to estimate the covariance between the offspring's phenotype and one of the non-transmitted polygenic scores. So this is arguably the most important covariance in the model. Like I was saying a second ago, it's kind of the crux of the whole thing. That's where we get the information on vertical transmission and genetic nurture. So you can start with model 0. And to get the covariance between YO and NTP, we just need to identify all of the paths between those two variables that abide by a set of path tracing rules. So here are all the path tracing rules. I will not go through them because this is a lot to go through, but I promise in practice, they're actually not too unwieldy to use. They're fairly intuitive. Okay, so for this covariance between YO and NTP, there are exactly two paths that meet all those path tracing criteria. So once those paths have been identified, we multiply the coefficients within each path and sum it together with the coefficients from the other paths, which sounds complicated, but it's not. So for this path on the left, we have 1 times F times 1 times half W. And for that coefficient on the right, we have 1 times F times delta times 1/2, which together simplifies down to half F times the quantity W plus delta. So mathematically, it's kind of like basic algebra, multiplication and addition, not too difficult. But one other thing I'd like to point out is that for both of these paths, note that they're going through the paternal phenotype. So in the case of no assortative mating, the only way that the non-transmitted alleles can have an effect on the offspring's phenotype is through their effect on the parent's phenotype, which makes intuitive sense. And that's actually one of my favorite things about structural equation modeling, is that these causal diagrams allow you to like visually see these different relationships between your variables. And it kind of is a sanity check, because we know that that should be the only way that the non-transmitted alleles can affect the offspring. So it's good to see that in practice. Okay, so that's the covariance for model 0. Now we can get that same covariance for model 1 that accounts for assortative mating. So as before, we'll trace all the paths between these two variables, and we'll start with the ones that do not use the copath. There are 1, 2, 3, and 4 such paths between the two. So note that 3 of these 4 paths are still going through the paternal phenotype. So that is still the primary way the non-transmitted alleles are connected to the offspring's trait. But note now that we have this path in red that does not go through the paternal phenotype. So with assortative mating, these non-transmitted alleles can still be associated with the offspring's phenotype through their association with the transmitted alleles. So there's an association there now that's totally independent of the paternal trait. Does that make sense? This is a lot. It's kind of complicated, a lot to keep track of. So if anything comes up, you have any questions, feel free to ask. Okay. So those are the 4 paths that do not use the copath. So now if we look at the paths that do use the copath, and thus that go through the maternal phenotype, there are actually 36 of those. Like 2 years ago, I sat down for a full day and just counted them. And I still think back on that day all the time. It was rough. And as you can imagine, having to identify all of the paths and all 36 paths, and then having to individually multiply all of the links within each chain and sum it together with all the links from all the other chains, that is not only super tedious, super laborious, but it's also really error prone. Take it from me. And I think for that reason, people get turned off from using asynchronous.
What are all of these paths? This is the maternal.
So these are all the paths between YO and NTP that travel through the maternal phenotype. So you can think about it like one path might go YO to TM through that way, one through the F, one through the, and so there's a lot of different ways that they can go through here, go around.
Yep.
There's
So there are no programs that you can put within your system better.
well, there are programs, we use OpenMX, for instance, that can help with the estimation, but you need to supply the algebraic expectations for your covariances. So this is an observed covariance. We have an observed offspring phenotype and observed PGS. So we know the true value of that in our data. And it compares that true value to the algebraic expectation that we're supplying. So fortunately, we've done all this work, so people who use this model don't have to do it themselves. Okay. So yeah, that's a lot, but fortunately, all of that work and mental anguish of identifying these different paths and adding together their coefficients can be avoided. So rather than doing that, we can instead just implement some shortcuts. For example, we can say that omega is equal to all the paths between YP and NTP. So that's the covariance between the internal phenotype and his non-transmitted PGS. And additionally, we can say that VY is equal to all the paths connecting YM to itself, so which would give us the variance for that phenotype. So two fairly simple shortcuts, but by implementing just these two, all of that math from all those different paths sums down to this equation on the bottom right. And it really, really cuts down on the amount of work, and the really helps prevent kind of easy to miss mistakes. So in short, I think that the math for our model one that accounts for assortative domain is not really all that much more difficult than the math for our model that doesn't account for assortment. And also note that the actual impact of assortment is being captured in those mu and g variables in the bottom right equation. So mu is part of the copath coefficient, and g is that covariance on the side. So if you were to use model one and there was actually no assortative domain for your trait, mu and g would be equal to 0. And if those two are equal to 0, then that equation on the right will be equal to the equation on the left. So which I think is cool. And again, another sanity check. Okay, so now that I've gone over how to estimate assortment, we can begin to address some other questions like what to do if the polygenic scores explain a small fraction of total heritability. And I want to emphasize this is something that I'm going over a little bit quickly, but that I hope that that brevity isn't taken as an indication that this isn't one of the more important pieces of the model. I think it's actually one of the most important things that it does, and one of the biggest advantages it has over past designs. So if we again start with model one, which assumes that this model assumes that the polygenic scores are accounting for the full trait heritability, which is an assumption made by past designs and an assumption that is wrong for pretty much everything, very wrong for pretty much everything. Most polygenic scores explain a small fraction of total trait variance and total trait heritability. However, we can relax that assumption by adding in latent genetic effects. So these latent variables are representing the proportion of additive genetic variance not captured in these polygenic scores. But that said, they operate really similarly to the polygenic scores I've already described. Like the observed PGSs, they have direct genetic effects on the phenotypes. In the case of genetic nurture, they'll covary with the familial environment. And in the case of assortative mating, they'll covary with each other and with the observed PGSs. So this is model two. And I understand that visually it's a lot to take in. It looks pretty gnarly. But I promise, not to sound like a broken record, but I promise that mathematically it's actually similar in complexity to model 0 and model 1. And that's something that we describe in our paper. We show how that's the case. A lot of the math from the previous models carries over to this model. And
sorry.
Okay.
With the late genetic facts, so there's been the Cohen and others very recently have these papers out saying you could sort of estimate that full genetic effects by sort of randomly taking GWAS and taking one part as an instrument for the other, and therefore that's I think the way instruments were designed that would sort of multiply that would produce the overall estimate of that. Brilliant, but they don't recover heritabilities, which are similar to twin heritabilities using that. I mean, does this approach really recover the full
Well, I hadn't read that paper, but that's interesting because that, if I'm understanding, is very similar to what we're doing. But our estimate of heritability, that's actually what I'll talk about now. So in this model, you can see that we have observed parental phenotypes.
Yeah.
So our estimate of heritability is coming from the degree of similarity between the parental phenotypes and the offspring phenotype. So we're looking at that level of similarity and determining how much of it can be accounted for by the observed polygenic scores. And the remainder is going into these latent variables. So for model 2, we do recommend that you have observed parental phenotypes. But if you don't, you can still use the model. You just would need to make an assumption about what the heritability is in your population of interest. So does that answer your question?
Yeah. Yeah, so you're assuming, but you're assuming that the resemblance of the parental and offspring phenotypes is all good. It gives you a reasonable estimate of heritability. Is that right? You use
Yes.
that to give you an okay.
Or if you prefer, you can use twin heritability estimates for this. But
No, that's doing something.
okay,
I'll send you the current thing.
that I would actually be really interested in reading that.
I thought it was genius to do it. Like, it's work.
That's what, yeah.
Well, definitely probably.
And I will say, I'll get to this in a little bit, but I mean, we've tested these models in simulation, but we haven't yet been able to apply them to real data because we haven't had a sufficiently powered one. But again, that's something I'll get to in a second. Okay. So in summary, we have created an accurate model of vertical transmission and genetic nurture, which again, I'm biased, but I think is really, really cool that we can accurately get an idea of just how much of an impact parents are having on their children's phenotypes. And this model can account for assortative mating at both equilibrium and disequilibrium. So that means assortative mating that has gone on for a really long time, assortment that has gone on for 0, 1, or 2 generations, things like that. It can also account for assortative mating that's due to phenotypic similarity, genetic similarity, or environmental similarity. Whereas most existing models, the vast majority, if they do account for assortment, have to assume that the assortment is due to phenotypic similarity. In addition, we can provide unbiased estimates of additive genetic variance, vertical transmission, and genetic nurture using only a polygenic score. We can easily extend this model to various types of situations to fit the needs of your trait and your data by virtue of these being structural equation models. And that last one is actually another one of my favorite parts of doing this. I've given this talk a few times, and sometimes people will ask, like, this is the data I have, how can we make the models work for that? And it's really fun to play around with the different pathways between the different variables to make it fit the question of interest or the data set of interest. So maybe you have grandparents in your data, we can make that work. Maybe you have cousins or something and you want to find a way to implement those, we can try to find a way to make that work. So if y'all have any data sets that you'd like to apply this to, let me know and we can figure something out. And another one thing I forgot to mention about us using structural equation models is that allows for direct comparisons between this and some of the traditional designs used in behavioral genetics, like twin family models or adoption studies, which I think is another strength of the approach. And normally I would want to talk about results here, but as I just mentioned, we don't have any. We'd hoped to apply this model within the UK Biobank, but unfortunately, the UKB only has about 600 trios and not enough parent offspring pairs to give us sufficient power to estimate what we want to estimate. So for the time being, we're working on ways to boost that power. But for right now, instead of results, I will talk about caveats and limitations. The first of which is that the model requires a large sample size to achieve equivalent power. So for a sample of for a trait with a PGS R squared of 0.05, you would need a sample of about 10,000 trios, which, like I just mentioned, is roughly 20 times the number that are in the UKB. And that's actually a big reason why I'm here. I'm meeting with Neil and others to talk about applying our model within the Norwegian mother, father, and child cohort study, or MOBA, which has about 60 to 70,000 trios in it. So that data set would give us enough power to estimate even traits with really small PGSR squareds. And if it's of interest, we do have a separate paper with Kim Kim as the first author, in which we detail how the power and our estimates vary as a function of sample size and PGS predictive ability. Okay. So in addition to that, we can only look under the lamppost, so to speak, at traits with sufficiently predictive power. So typically, like rule of thumb is a PGS R squared of about 0.1, 0.01 or 0.02, which a lot of traits do have PGSs that are that predictive, but for more niche things, we'll have to wait until we get larger GWAS of them. Third, our models are only capturing the effects of a single parental trait on a single offspring trait. And we view this as both a strength and a limitation, because while it's only offering a sliver of the full picture of parenting, having an understanding of which specific parental traits lead to different things in the offspring is really useful and important information. If you're able to say trait X, Y, or Z leads to depression or alcoholism in the child, that's actionable information. And that opens the door to different pathways for intervention, which I think for me and for probably everyone here is sort of the ultimate goal of our research. And that said, if you are interested in knowing the full effect of all parenting behaviors on an offspring trait, there are models for that, like Trio GCTA, for example. And I think the name of the game with that is just kind of triangulation of results across different methods. Okay. And finally, this model does not account for the effects of collateral relatives like siblings, which, as I'll mention in a second, is something that we're currently working on. But as one quick, oh yeah.
Joe, one question. So a key parameter of interest for the group here is about the causal effect of one phenotype on another. So presumably from the path diagram, it's pretty easy to back out the effect of, say, the parental phenotype on the offspring phenotype, i.e. not just the effect of the non-transmitted allele. If you presumably it's just F is a parameter that's identified in the model. So you
Vertical transmission, yeah.
You can just report that and have that as your effect estimate.
Well, actually, I think the more informative statistic would be this VF here, the variance of these familial environments, which gives the proportion of the phenotypic variance due to vertical transmission.
Yeah, so in some ways it depends on the audience, right? Because some of the audiences that we're speaking to are going to want to know if we are intervening on this phenotype here, what's going to be the effect on the outcome of interest. So they're less used to just working in terms of variances. So if you've got the if you can report the effect on the scale that they're used to, it's quite it can be pretty persuasive one.
Oh, okay.
Particularly for clinical and policy audiences.
Okay, because I work with a lot of people doing twin family research, and they think entirely in variance components.
Yeah,
So
yeah, which is completely intuitive to them. But for kind of outside that community, it's I think that's one of the strengths of doing randomization is that it does get that parameter that is very intuitive for people because it's just what everybody else is working with.
We just need everybody to start working with this first, and then we'll be there.
Well, it's almost a case of pulling out your estimate and presenting it to everyone in a very, very familiar way. And then the back end and all the calculations that you're doing, they don't necessarily need to get into all of that as long as they can interpret, oh, this is that on that.
And that is a hot topic of debate among our team about what the best way to present, what the most intuitive way is. I mean, you know how it is when you've worked on something for years, you kind of forget what it would
depends
like
on
fresh eyes. Yeah,
it depends on the journal and the audience. Yeah,
exactly.
In a massive way.
And I think that gets at the purpose of this model can be one of two things: either to look at the effect of parenting if that's of interest, or to quantify it so that you can remove it from other estimates and other studies. Cool. Yeah, any questions before I go further?
Sorry, you're talking earlier about a situation where you had different traits in the parent. Okay, and so is that saying that? So you're talking about why P and YM here being a different trait measured in the parent compared to the YO in the offspring. Is that?
So I might have been unclear. For this model, we can't do that. I mean, you can plug in other phenotypes for the parents than for the offspring. And past approaches have done something to that effect, but then you're not accounting for within trait vertical transmission or cross-trait assortative mating, because these two are the same and these two are different.
Yeah.
Does that make sense? So the results will be biased because of that.
So with this one, you're saying you're talking about education and parents of education.
Yes.
And hence you've got the same little delta across. All of
Exactly,
this.
exactly.
Yeah,
But that same delta does get an important point, which is that I forgot to mention in the limitations. We are assuming that the PGS is equally predictive for the parental generation as the offspring generation.
Yeah.
Which is potentially a big assumption and one that we're trying really hard to figure out a way around.
You've also got symmetry between the left-hand side and the right-hand side, haven't you?
So no sex effects.
Yeah, yeah. So that would be both in terms of genetic effects and the effects on the offspring.
Yeah, exactly.
So you'd be ruling out any sort of introducer-line effect or socially.
Exactly, yeah. And there is a lot of evidence that children tend to be more similar to their mothers, but also mothers typically fill out children's questionnaires. So there's also that kind of reporter bias. So it's hard to know how much to take that with a grain of salt. Sure.
The question about, because obviously I'm guessing this model also assumes that all the phenotypes are similarly, as in like there's no measurement error for the phenotypes as well. So if you have lots of, because you do have the epsilon, so maybe you do account for
Yes.
measurement error.
Yes. Yeah.
So it doesn't really matter, I guess.
Hopefully. Ideally, yeah. And simulation, it does not.
Okay.
Okay. Well, I can't talk about any results from this full model. I can say that we have looked at just the top half of the model, because the UK Biobank actually has almost 70,000 spousal pairs in it, 70,000 mate pairs. So we can remove out the vertical transmission components and just look at the assortative mating components. And that in itself can give us a lot of really cool information. For example, whether the trait's being assorted upon, whether the magnitude of assortment has changed continuously over time, and whether it's being driven by phenotypic, genotypic, or environmental similarity between partners. And as you might have noticed, without a child, you can't divide things into transmitted and non-transmitted, because you can't transmit something to somebody who doesn't exist. But so in that case, we just divided the parental genotypes into two random halves, so that we can still compare the within-person covariance to the between partner covariance. And I'll go over this pretty quickly because it's sort of an aside, but we've done this for about 30 traits in the UKB, and we found a wide variety of patterns of assortative mating. I will say these results are kind of preliminary, so take them with a grain of salt, but suffice to say, for some traits like educational attainment, for the past century, we've seen assortment increasing over time, such that mates are becoming more similar to one another. Whereas for things like height, the assortment seems to have been continuous over the past century. Meanwhile, for other traits like schizophrenia, there is no evidence of assortative mating on the liability to schizophrenia and a lot of other traits. Meanwhile, for some traits, like for educational attainment, we see that the genetic correlation here in red is a lot greater than what we'd expect based on their phenotypic correlation, suggesting that mates are choosing one another based on genetic similarity, which is a little bit of a weird thing to think about, but it suggests that they're actually mating on a genetically correlated trait, say maybe intelligence or conscientiousness, rather than actual educational attainment.
Could that be a measurement thing? So if your education categories are fairly coarse, and you've just went to university or dropped out of high school, then if they're matching on something that's much more fine-grained than that, for example, went to Oxford University rather than Bristol University, then you would get results like that.
I think that's possible, and that this is something that we've been trying to tease apart to make sure that it's real, because we've actually seen this pattern for, I think, most of the assorted upon traits in our sample, so that's a possibility. I think that with that specific case might fall under the category of environmental similarity, though, wouldn't it?
Yes, potentially.
But I'm sure there is an example you could come up with that would get at what you're saying. It gets a little tricky really quickly.
It's not intuitive, right? Because you'd expect phenotypic similarity to be, that's what they're matching, right? I mean, it has to be something phenotypic.
Well,
Because it can't see the genotype.
in this, yeah, exactly. In this case, it would be phenotypic mating, but for a separate genetically correlated trait, if that makes sense.
Yeah.
Yeah, so say intelligence. So the assortment on educational attainment is taking place to the extent that it's genetically correlated.
Yep.
So yeah, again, not to get too bogged down with this because this is an aside, but we're hoping to have this out in the next couple of months, and something I'm very excited about, so I wanted to mention. And lastly,
Is this from the UK Bio?
yes,
Yes, so I mean, one thing to think about there is that because it's sort of mating leads to genetic correlations across chromosomes, etc. Which is it's really interesting. UK Biobank's 5% sponsor a volunteer sample. And schizophrenia contract scores is the strongest predictor of participation or non-participation in UK Biobank. So it becomes associated, so you get cross-chromosome correlations for anything which doesn't have a perfectly log additive joint effects on participation. So so if you've got high schizophrenia score to get into UK Biobank, you'll have a higher, you'll have the high schizophrenia score lowers your participation chance. So you generate correlations with factors which lead to participation in UK Biobank.
yes,
I mean, it's a devil's advocate point of view. In fact, 5% participation is generated as much association as it's a sort of maybe.
I
haven't thought schizophrenia might have that because what if you have the whole population, you might show a sort of maybe on the schizophrenia score, but by the time we get to Biobank, the fact you have which gets you into Biobank despite having high schizophrenia score, they're all not correlated, they're all different chance things which have got you into one. It does screw, it screws the participation of Biobank, does screws things up. Like as you know, your GWAS age and you get those bits for genetics causes age and you go Biobank. Autosomal genes cause sex and causes sex, etc.
yeah,
So
we've seen plenty of weird things.
It's weird. It's a weird sample we're doing. We're trying to estimate population parameters.
I wish I could disagree with that, but I cannot. Yeah, I think it was really important us to use it in the UKB to demonstrate its accessibility of the approach, but we are having to kind of figure out how to explain some weird stuff.
Yeah.
And it's a lot of, is this us? Is this them? What's causing it? Educational attainment is super weird in the UK Biobank. It follows a lot of weird patterns.
Yeah.
Traits that you would expect to have really high PGSR scores have really low PGSR scores in the UKB. And that's really messing with what we've got going on here.
So one thing I've been talking about is that we're really interested in compare when you've actually got the Offspring transmission data, we're interested to compare what that predicts, outcome things that you can associate it that directly with what we see in UK thing. I mean, mode is 50% response rate. So it's way better, but also, of course, you've got a wonderful data. You've got a wonderful situation of being able to look at transmission. So some of these things might be a bit different. Start looking at them in a sample with that.
Has Mova got an issue that it's parents? Because I mean,
Yeah,
that's selecting on having an offspring.
91%. So 5%.
Is that true for both males and females?
So it's I mean, it's less selective, obviously, 5% volunteer.
The mobile recruitment was through fertility and like OBGYN, so it's correct.
50% of all women who became pregnant over that area got it, except for some tiny outlines and
Okay.
some real tiny outlining. Areas. And it fluctuated up now.
Sure.
Uh,
Well, I'll be in Oslo in a couple weeks to meet with them about this, so maybe I'll see you there.
Are you going for that meeting that moment?
I don't think so, but
September.
I leave the 27th.
How about?
No one told me. Okay, well, I'll try not to take that personally.
It's just that they have it, it's just that one makes it happen. You don't get it.
Sure, yeah.
It's more.
Mhm. Yeah, and we might reach out to you later on once we get a better sense of these results, because I think we're beating our heads against the wall a little bit with some of them. Well, to close out, talking about some future directions. I had mentioned trio, spouse, and parent offspring models, how we can use models that are with some missingness and be okay. But we're also working on leveraging sibling data, because there are a lot of siblings in the UKB, and it would be great if that could boost us over the edge of having sufficient power to estimate things in the UK Biobank. In addition, and this is what you were asking about earlier, we'd like to do things multivariately. So to look at the effect of a parental trait on a different offspring trait. And we're fairly certain this works, that you can substitute all those scalars with K by K matrices, which would allow you to do that. It would account for within and cross-trait vertical transmission and assortative mating. And hard to believe, but path tracing, like I did earlier with matrices, is actually no more complicated than it is with scalars. Pretty much all of the math from our models transfers over to the bivariate case, which is fantastic. And as was also mentioned, we'd like to be able to account for parent and offspring sex effects. So being able to differentiate father-son, father-daughter, mother-son, and mother-daughter effects from one another is a big priority. We think that'll be really interesting. We're also working on isolating specific parenting behaviors, which will be especially great with the MOBA data, because they have really, really fine-grained measures on specific parenting traits and behaviors, like how stringent their parenting is, how often they lose their temper, how many rules they have, things like that, how they behave in front of their offspring. So in doing so, hopefully we'll open the door to more clear environmental interventions. And lastly, we'd like to apply our models within a more diverse sample than the UK Biobank, both more culturally and racially diverse, ancestrally diverse, but also, that just have a larger range of outcomes, larger variances. So in addition to MOBO, we're also working on getting data from the environmental influences on child health outcomes, the ECHO study in the US, which will allow not only for more generalizable results, but for cross-cultural comparisons. So with that, I'll thank my co-authors, Youngdong and Nat, our funding sources, my dog, and I will take any questions that you'll have for me.
Thank you very much, Jared, for a wonderful talk. I will open the floor for questions. I'm aware that it's past and people may have somewhere to go, but if there are some final questions. Okay.
Yeah, I was going to ask a very quick question about your models. I mean, so I was just kind of thinking about like gene and environment interaction. Like, I mean, like if you're like throwing in a couple different things, how it works. Like, have you guys thought about looking at that, or really keep it simple as that?
We've talked about it, and at the very, very high level, we think that that's something that could be incorporated down the line.
Okay.
Currently, we are not accounting for that. I think one way we thought about it also is like those bivariate models. You could put the moderator, the environmental moderator, in there somewhere, and that could maybe get at it. But yeah, we haven't figured out the solution quite yet. I haven't thought about it too hard yet.
Presumably with the multivariate models, there's an issue with power, right? As soon as you start adding in additional phenotypes, your data requirements are going to be inflated.
Theoretically, yes. Can't say for sure yet.
It'd be interesting to know how that works out with the MR, whether we've been doing with multivariate models. You run into issues of power very, very quickly. So you can kind of do 2 exposures, but anything more than that, and it's challenged very, very quickly.
Okay.
Even with enormous numbers.
Okay. Because I think so far we've only looked at it within simulated data that, you know, we've by design and intentionally made it very, very well powered, you know, 100,000 families or whatever, just to make sure that the models are working as designed. But I don't think we've actually looked at bias changes.
Yeah.
I'm sorry.
That's all.
No, that's okay.
You're not finding the room.
Thank you.
Thank you.